let's talk about the biological macromolecules that will be involved in this biochemistry class. A lot of this is going to be review from previous classes you've had, or maybe even high school biology. So we're just going to go through it quickly, bring that knowledge back up to the forefront of your brain. So when we're thinking about biological macromolecules, we're thinking about proteins, nucleic acids, lipids, and carbohydrates. Those are the four macromolecules that are the building blocks for life. So we're going to quickly step through each of these, remind you of the basic structure, the chemistry of them, and how they're held together. And this is really interesting. First, thinking about the elements that make up life. So this table is showing different elements and what percentage that element makes up a human body. So we can see that we are about 63% hydrogen. And then this is interesting, comparing that to seawater. So we have about the same amount of hydrogen as what is found in seawater. A little less oxygen found in seawater. And why is that? Because there's more carbon in our bodies. So we'll see that carbon is really the building block for all of these biomolecules that we're going to be looking at. Nitrogen is there too, and then we also have some of these ions that are going to be important for biological molecules such as enzyme activity. Phosphorus, of course, is built into the head group of lipids, and that kind of thing. So there's this joke about the organic chemist or the biochemist periodic table of elements where only these atoms are shown on that periodic table. So we're really just going to focus in on those, forgetting about a lot of the other elements that came up in your general chemistry classes. All right, so starting with proteins. So proteins are made out of amino acids, that's their building block, and those amino acids are covalently bonded together. So where each one of these colored beads on a string, each one of these beads is an amino acid, and it's covalently bonded to its neighbor, and these make a long chain of amino acids. So this is the primary structure of the protein, are those covalently bonded amino acids. This primary structure, this long chain of covalently bonded amino acids, then folds into a secondary structure, primarily alpha helices and beta sheets. I know you guys have seen these before. They're going to come up again in more detail when we get to proteins. These secondary structure elements are stabilized by hydrogen bonds, so that's a non-covalent interaction, much weaker type of interaction, which is important because that makes this reversible. And then these secondary structure elements fold into a tertiary structure, where these alpha helices and beta sheets interact with each other to give the protein its three-dimensional shape. And then for some proteins, we'll have multiple subunits, multiple polypeptide chains that assemble to form a quaternary structure. So again, everything after this first step is driven by non-covalent interactions, such as hydrogen bonds, hydrophobic interactions, charge interactions, to make the protein fold. There are 20 amino acids. We are going to go into these in more detail, and you've probably seen these before, so I'm not going to spend too much time on them now, but just know that there's 20, and eventually we're going to be spending a lot of time looking at the structure and chemical properties of these 20 amino acids. When we think about proteins, here are a couple examples of why proteins are important in the cell. The first example is that proteins can be structural. So for example, the cell's cytoskeleton, which is like the bones of the cell, are made out of proteins such as actin and microtubules. And in the cell microscopy image, the microtubules are stained green and the actin is stained red. So these proteins are just structural. They give the cell its shape, they help it move, they are structural. Another example of key protein function in the cell are signaling proteins. This includes, for example, membrane receptors. So if here is the cell membrane or the plasma membrane, we can have a transmembrane protein that receives signals from outside of the cell and then finds a way to pass the message inside of the cell to start a downstream cell signaling cascade. We'll talk a lot about that later in the class as well. And this diagram is just showing the pathway for adrenaline signaling. And then a third example are enzymes. So enzymes are proteins that are doing work. When I do this activity with, this outreach activity with elementary and middle school kids, I call enzymes the builders and breakers in the cell because they are taking some other chemical and breaking it down or building it up. So here's one example of an enzyme that converts maltose into glucose. So the substrate for the enzyme, the thing it's acting on, will bind in the enzyme's active site, and then the enzyme acts as a catalyst to speed up this chemical reaction. Enzymes are also important for building all the biomolecules in the cell. So, for example, building those amino acids into a long polypeptide chain, covalently linking them. So when we're seeing changes in covalent bonding, like what's shown here, it's oftentimes an enzyme that's the force behind that, speeding up that reaction. All right, now moving on to nucleic acids. So again, you know this, you've seen this before, but I just want to give a quick reminder. So we know that there are four nucleic acids here involved in the structure of DNA. They are thymine, adenine, cytosine, and guanine. Remembering that it's hydrogen bonding that makes the specific base pairing, why thymine pairs with adenine, so T with A, C pairs with G because it forms these three hydrogen bonds. Remembering also the basic structure of DNA where you've got the base here, so thymine, for example, connected to a sugar group here, connected to a phosphate. And for DNA, we have a hydroxyl group at the 3' site of this ribose sugar. So that's the basic structure of DNA, the phosphate sugar backbone and the bases that are bound to it. And then those two strands of DNA interact with each other through those non-covalent interactions, those hydrogen bonds. So this is what our double-stranded deoxyribonucleic acid, or DNA, is going to look like. And then remembering we also have single-stranded RNA, which is used in changing that DNA into protein. And we'll talk more about that. One other really important nucleic acid I want to bring up now is adenosine triphosphate. So by the way, what's the difference between adenine and adenosine? So adenine refers to just this base here, just this part you see in orange, whereas adenosine refers to the whole base sugar phosphate complex. When I say adenine, I mean just what you see in orange. When I say adenosine, I mean this whole part here. So when I say adenosine triphosphate, it's that same adenine base plus the sugar plus 3 phosphate groups, so triphosphate. This is a free nucleic acid that is used not just for production of DNA and RNA, but also as an important molecule for energy storage in the cell. And when this third phosphate gets cleaved off, it releases energy that the cell can then use to power other cellular processes. And we're going to talk about this a lot, but I just want to introduce it now so you've seen it. This is ATP adenosine triphosphate. Our third biological macromolecule are lipids, or phospholipids. So phospholipids are composed of that hydrophilic head group. So we can see a phosphate and a glycerol backbone here. These are all hydrophilic. And hydrophobic tails. So these are carbon-hydrogen chains, hydrocarbon chains. Sometimes they have double bonds, which give the tails a bend to them, these unsaturated fatty acids. And this section down here is going to be hydrophobic. So because we have the hydrophilic head and the hydrophobic tail, it makes these phospholipids self-assemble into lipid bilayers that look something like this, where the hydrophobic tails want to interact with each other and get away from the water that's on either side of the membrane. And then the hydrophilic heads, they like to interact with the water. So this makes this nice sandwich structure that is forms chemically favorable interactions. And then last, we have the carbohydrates. So carbohydrates generally have this kind of ring structure, either 6 or 5 members in the ring, one of them being an oxygen. And then we'll have different hydroxyl substituents with different chiralities, so pointing up or down, depending on the position. And there's very specific naming for all of this that we'll get into later. So a carbohydrate monomer is going to be one of these sugars. This is glucose, for example. This is fructose, for example. Those monomers can be assembled into larger disaccharides. That's what you see here. Sucrose is a disaccharide made of a glucose monomer and a fructose monomer covalently bound together. And you can also covalently bond these into larger polysaccharides, which we'll show on the next slide. So carbohydrates are commonly associated with energy storage. That's where we're going to see them come up a lot in this class. So we talked about those polysaccharides. One that's very important is when you take glucose, and it can be stored as glycogen in the cell. So glycogen is when you have many, many glucose molecules covalently linked together. So you can see each one of these is a glucose. And when we link a bunch of them together, we form this larger molecule called glycogen. And glycogen is a way of storing energy. So then that glycogen can be broken down to make glucose. And then why is that important? Because glucose gets broken down to make ATP for the cell in the process of glycolysis, which we'll talk about a lot. So a practice question, you can work on this on your own. We're also going to talk about it in class. So considering proteins, nucleic acids, and lipids, what intermolecular interactions drive their macromolecular structure? Or another way of thinking about this, after the covalent bonds are formed, what non-covalent bonds hold these structures together? So that'll get you to start thinking about the chemistry of these structures and how their chemical structure drives their macromolecular structure.